amino acid 1 is attached to amino acid 2, building an amino acid sequence. Let me just probe a little bit. So, assuming you've had a transcription and a translation event, and you've made a protein. You guys have been through this in your life. Don't tell me you haven't. What happens to this growing chain of amino acids as it is constructed? Help me know that you guys are on board with me. As it grows, what happens to it? As this amino acid chain grows, what happens to it? I'm building a protein. What?
Folding.
Okay, it starts folding. And depending on how long the thing gets, it can enter into a secondary structure. And there are two common secondary structures. They would be, it's not on your presentation, I'm just probing from where you've come from.
Alpha helixes and beta sheets.
The alpha helix and beta pleated sheets. Okay, if the protein is larger, it can take on a more complicated structure than a secondary structure. Most of the time caused by hydrogen bonding and the folding of this thing. And as it becomes a more complex structure, we say the protein now has more than secondary structure. It has tertiary structure. And sometimes proteins that have tertiary structure within cells are linked together with other tertiary proteins to make a protein that has even a more complicated structure, and we would say that it is, it's the last one.
Quaternary?
Yeah, quaternary structure. So proteins, depending on how big they get, can take on these very sophisticated folding patterns to become functionally significant in the cell. And that's all molecular. That's very cool. Now, what I want to do with you to finish this is to go back one more time in your lives and talk about how these proteins were constructed. I have tried to make the case over the last 35 minutes to you how important proteins are to anatomy. So at this point in your life, I'm hoping if somebody says, what's the most important organic macromolecule in explaining anatomy? You would say, it's not lipid, it's not sugar, it's not carbohydrate, it's not even nucleic acid. What's the most important protein? What's the most important molecule? Proteins, yeah? So if I'm going to explain the insides or the outsides or how they're linked together, how they interact with each other, the different kinds of tissues, the different kinds of cells, all of that is the foundation of all of that is in the nature, the structure of the proteins that are there. What is the collagen and the elastin and the actin and the myosin? What do they look like? The tubulin? Okay, so I'm going to finish this by just a very, very sketchy kind of, this is where they came from. And y'all can help me, right? When I say a gene, people talk about genetics and things are passed, traits are passed on, and you look like your mom and I look like my dad kind of thing. What do you mean in science when you say a gene?
DNA.
Okay, so we're talking about the DNA. So here is the base structure of DNA, which is very simple compared to proteins, right? The DNA is a ladder, a double helix, where the outside bars of the ladder are made up of repeating sugar phosphate groups. Repeating sugar phosphate groups. And the sugars here, deoxyribose in this case, is attached to one of 4 nitrogen-containing bases. They are adenine, thymine, cytosine, guanine. Okay, so these 4 nitrogen-containing bases. And we know that in the structure of the DNA molecule that cytosine will always base pair, form hydrogen bonds with its counterpart, guanine. And adenine will always base pair with its counterpart, thymine. So when you unravel the DNA, when it is replicated in cell division, we don't worry so much, although you can make some mistakes. We have mechanisms in place to help repair those also. But if you unravel the thing, you know how it's going to look when it gets reconstructed. It's called semi-conservative replication. I know that if I cut this thing in half, the nucleotides that come in here to rebuild the new one will be the same as the original. Guanine always goes with cytosine, adenine always goes with thymine. Okay, but you still haven't told me what you mean when you say a gene. So you said DNA. So you're telling me that genes are DNA.
It's embedded in the DNA.
Say again?
It's embedded in the DNA. It's embedded in the DNA.
It's embedded in the DNA. Okay, so in a typical human cell, there are 3 billion base pairs in a single double helical strand of DNA. So it's a lot of them. And you're saying the genes are embedded in that. Yes? Okay. In what way? How? How do you mean embedded in that? If you were going to explain genetics to me, the base of genetics, how would you do that? Get me started. Take me from this. What part of this would be a gene?
It's like just a certain combinations of them build up into form genes.
Okay. Help me. There would be a sense strand and there would be an antisense strand, right? What do you mean by sense and antisense? I know I'm probing. It's not really my job to teach you this, but I'm going somewhere with this. So what do I mean by that, Jared?
So don't like every 3 of those code for an amino acid, and then certain amino acids code for like the end or beginning of a section, and that makes up like a gene?
Okay, so we're getting closer. So he's talking about codons, 3 base sequences that code for specific amino acids. We're going to do that on the next picture. But I want a bigger picture here. This is a good thing about anatomy. You're going to love this. Trust me. In molecular and micro and these other classes, you get to tease out the little bitty details. Here, this is big picture. I want to know where the proteins came from. They came from this molecule. And making the proteins is, in fact, what we mean when we say genes. We mean that some part of this molecule has been somehow used or given instructions to the cell on how to make a protein. That is what we call in science the central dogma. The central dogma is this. Parts of the DNA molecule will unravel. Enzymes do this. So that one side of the DNA, the sense strand, right, will be copied into a new single-stranded molecule that we call RNA. Am I doing okay? And that RNA will then serve as the messenger to tell the cell how to make a protein. Now, that section of DNA that is unraveled and copied, the sense strand, is the gene. So, fundamentally speaking in science today, a gene is a part of the DNA, piece embedded in it, in fact, one side of it, that is then used to give instructions to the cell for building a protein. That's what we mean, really, when we say the heart of genetics. And this shows it to you. So here's the DNA. It's been unraveled so that I have one part of the DNA, the coding strand. And there's the base sequence, TACCCG, AGC, etc. Those are the bases. This DNA coding strand, one side of this thing, is this purple line is now the gene. And the gene then is going to be copied. This copying takes place in the nucleus. The single-stranded molecule that this gene is copied onto is called the messenger RNA, and it is copied in the same way the DNA would be copied in replication, in a semi-conservative fashion. That is, A always goes with T, C always goes with G. The big difference in the bases is, where there's an adenine on the sense strand or the coding strand, you're going to get a uracil on the messenger RNA. There's no thymine on RNA. So the base sequence for the messenger RNA, if you know the base sequence for the DNA, is very predictable. You know what the bases will be. And you all will have to learn, if you didn't have to learn them in organic, you will have to learn the codes for the 20 amino acids that are used to make the proteins of life. We call those the codons. And they are 3 base sequences specific for particular amino acids. So TAC on the DNA strand gives rise to AUG on the messenger RNA, which will always code for methionine. That is normally the first amino acid for any protein made in the human body. It is arguably the most famous codon in anatomy. AUG gives instructions for methionine. The next 3 base sequence here, GGC, codes for glycine, and on we go. So there are 3 base sequences then that are used to tell the cell what amino acid goes next in the strand. Now, does it matter that methionine goes first, glycine goes second, serine goes third, isoleucine goes fourth in this particular protein? Will it matter that the codons here code for these specific amino acids? If you do not put the right amino acid where it goes, what happens to the protein?
Changes.
Okay, in what way does the protein change? So when we're building, a minute ago, I said we're building the protein, and as they build, they can go through structural changes as they begin to interact. They go through secondary structures, tertiary structure, quaternary structure, depending on how long the chain is, and depending on where all the side groups are, where all the hydrogens and the R groups are. It matters, right? Okay, can you give me some human examples of what it would mean to put one of these amino acids in there? You know, if there was a genetic mutation that caused the wrong amino acid to go there. Any examples of this in your life that you've heard of?
Like cystic fibrosis?
Yeah, so cystic fibrosis is caused by a dysfunctional chloride ion channel in the membranes of the pleura of the lungs, so that you don't transport charged ions and the water properly, and it causes thickening, mucus to build up within their lungs. This is caused by a genetic mutation, and we know where it is now because of the Human Genome Project. The question about how to fix that is another question. That's a problem.